Let's microbiology it by micro mind mapping. This will allow you to retain the basic information before you open the textbook and go to a lecture. Our core is external structures. Stemming from that, we have glycocalyx. That's our first branch. From there, we have capsule, slime layer, and EPS, or extracellular polymeric substance. Our second branch is flagella, and it's made up of filament, hook, and a basal body. Our third branch is fimbriae, which are small mini appendages that is used for attachment. And our last, fourth branch is pili, which is used for gliding, twitching, and conjugation. Prokaryotic structures external to the cell wall are critical for attachment, motility, environmental sensing, survival, and genetic exchange. These structures allow bacteria to interact dynamically with their environment rather than existing as passive cells. The most significant of these structures include the glycocalyx, capsule, and slime layer, flagella, fimbriae, and pili, each contributing to bacterial fitness, pathogenicity, and ecological success. The glycocalyx is a viscous, gelatinous layer composed of polysaccharides, polypeptides, or both that surrounds the cell wall. When the glycocalyx is organized and firmly attached, it is called a capsule. When unorganized and loosely attached, it is referred to as a slime layer. The glycocalyx plays a major role in the adhesion to surfaces, protection from desiccation, resistance to phagocytosis, and avoidance of host immune responses. A key function of glycocalyx is its role in biofilm formation. Biofilms are structured multicellular bacterial communities embedded within a matrix of extracellular polymeric substance, or EPS, which is largely derived from the glycocalyx. Biofilm formation typically occurs in stages: initial attachment to a surface, microcolony formation, maturation into a 3D community, and eventual dispersal. Within biofilms, bacteria communicate via quorum sensing, sharing nutrients and displaying increased resistance to antibiotics and disinfectants. Common examples include dental plaque, biofilms on catheters, and microbial communities on rocks in aquatic environments. Flagella are long filamentous appendages responsible for bacterial motility. Structurally, a flagellum consists of three main components: the filament, composed of flagellin protein, the hook, which connects the filament to the basal body, and the basal body, which anchors the flagellum in the plasma membrane and cell wall. The basal body functions as a molecular motor, allowing the flagellum to rotate rather than whip, propelling the bacterium through its environment. Bacterial movement is guided by taxis, the directed movement towards or away from environmental stimuli. Chemotaxis allows bacteria to move towards nutrients such as glucose or amino acids and away from toxic substances. Phototaxis occurs in response to light, particularly in photosynthetic bacteria. Taxis allows bacteria to optimize survival by locating favorable conditions and avoiding harmful environments, and it is mediated through sensory receptors and signal transduction pathways. In addition to flagellar motility, some bacteria move using twitching and gliding motility, which are mediated by pili rather than flagella. Twitching motility involves the extension, attachment, and retraction of pili, pulling the cell forward in short, jerky movements across the surface. Gliding motility allows bacteria to move smoothly along surfaces without visible appendages, often through coordinated pili activity or secretion of polysaccharides. These forms of movements are particularly important in surface colonization and biofilm expansion. Pili also play a crucial role in horizontal gene transfer, especially through conjugation. During conjugation, a sex pili forms a bridge between two bacterial cells, allowing the transfer of plasmid DNA from a donor to a recipient. This process enables rapid genetic variation and the spread of advantageous traits such as antibiotic resistance, metabolic capabilities, and virulence factors. Horizontal gene transfer allows bacterial population to adapt quickly to environmental pressures without relying solely on mutation.